Hi everyone, this explainer will examine a recent study that uncovers the long-term heritable consequences of DNA double-strand break repair on genome function. We'll begin by addressing a fundamental gap in our understanding of genome maintenance. Then we'll detail the experimental model used, present the core evidence for persistent functional impairment, explore the underlying structural changes, and finally, introduce the unifying concept of chromatin fatigue. So we know that DNA double-strand breaks, or DSBs, induce massive and transient alterations in 3D chromatin architecture. This restructuring is a necessary step, of course, to facilitate repair by attracting the necessary machinery. But this raises a critical question. Once the DNA sequence integrity has been restored, are these extensive alterations fully reversed? Or do some lingering changes persist that might affect cellular function and even be inherited by subsequent generations? This research was designed to address that very gap. To investigate the long-term consequences of a single repair event, the researchers developed a system to induce one precise DSB. Their target was a specific, topologically sensitive region, the 2.8 megabase topologically associated domain, or TAD, which contains the CMYC gene on chromosome 8. They used Cas9 ribonucleoprotein, or RNP complexes. This ensures a rapid DNA cleavage without the prolonged presence of the enzyme. 12 distinct sites were targeted across the entire TAD, from within the CYMC gene itself to locations megabases away. This setup allowed them to assess how the location of a single repaired break affects gene expression. And the results were quite clear. A single repaired DSB causes a protracted and widespread impairment of gene expression, not just for the gene at the break site, but for others within that same topological domain. Here we see CMYC protein levels measured 24 hours post-transfection, a time point by which DNA repair is largely complete. Now, compared to the control, expression remains significantly reduced. A cut within the CMYC open reading frame led to a 60% reduction. And even when the break occurred 300 kilobases upstream, a 30% reduction in CMYC protein still persisted. So to determine if this effect originated at the level of gene expression, messenger RNA levels were measured. Analysis via RT-qPCR confirmed that CMYC mRNA was repressed in a pattern very similar to that of the protein. This finding indicates that the functional impairment is indeed happening at the transcriptional level. And this transcriptional dampening wasn't just confined to CMYC. The expression of other genes inside the same TAD, including the protein-coding gene POU5F1b and the long non-coding RNA PVT1, also showed sustained repression. This demonstrates a long-range effect of that single repaired break. Now, a crucial finding of this study is that this transcriptional impairment is heritable. The reduction in CYC expression wasn't transient. It persisted for up to 96 hours, a period that covers multiple cell divisions. This demonstrates that a memory of the DNA damage is propagated to daughter cells. This conclusion was tested directly through single clone expansion experiments. Cells that had repaired a DSB were isolated and allowed to divide. The resulting clones inherited the reduced CMYC expression levels. Not only that, but these clones also displayed a reduced dynamic range of CMYC expression, which suggests a loss of normal transcriptional plasticity. Okay, so having established a heritable functional impairment, the study next investigated the underlying structural changes in the post-repair chromatin that could provide a mechanistic explanation. Using dual-color 3D DNA fish, the physical distance between the two ends of the CMYC TAD was measured. 24 hours after a DSB, the median separation increased from 0.45 micrometers to 0.63 micrometers. This finding points to a lasting loss of compaction, resulting in a more decondensed, open conformation of the TAD post-repair. High-resolution region capture microSEQ provided an even more detailed view. The data revealed a persistent loss of long-range chromatin interactions that spanned the location of the former break site. This loss was observed up to 72 hours post-repair. And at the same time, there was an increase in local short-range interactions, suggesting a fundamental and lasting reorganization of the domain's architecture. These structural changes appear to correlate with impaired RNA processing. Using RNA fish, the study observed a sustained reduction in the local accumulation of both CMYC mRNA and the PVT1 long non-coding RNA at their transcription sites. This compromised RNA retention suggests the structural scaffold required for these local RNA compartments is disrupted. When you put all these findings together, The heritable transcriptional impairment, the lasting architectural changes, and the compromised RNA retention, they culminate in the proposal of a new concept to describe this state. The phenomenon has been termed chromatin fatigue. It represents a previously uncharacterized consequence of DNA damage that extends beyond just restoring the primary DNA sequence. You can think of it as a memory of the break, encoded in the structure and function of the chromatin, which is then passed down through cell division. And this chromatin fatigue has tangible physiological consequences. For example, when a post-repaired C-my-C locus was stimulated with epidermal growth factor, a mechanical driver of its expression, the transcriptional response was significantly blunted. This suggests the ability of gene regulatory elements to respond to signals is compromised. On a larger scale, the accumulation of such fatigued loci might contribute to the functional decline associated with cellular aging. So the proposed model can be summarized in a few stages. First, a DSB occurs. Second, the locus restructures for repair, which includes transient transcriptional silencing. Third, after repair is complete, the locus doesn't fully revert to its naive state. It instead enters this persistent state of fatigue. And finally, this process can repeat, leading to an accumulation of fatigued loci throughout the genome, which progressively alters cell physiology. The discovery of chromatin fatigue introduces a new dimension to our understanding of DNA damage and repair. It prompts critical questions for several fields. For therapeutic genome editing, it suggests that even a perfectly repaired cut could have unforeseen, lasting effects on neighboring gene function. And in the context of aging and oncogenesis, it provides a potential mechanism by which DNA damage accumulation leads to stable, heritable changes in gene function that can drive disease.